Hello, everybody. Welcome. My name is Vivian Cao, and I am the marketing coordinator at ASEA Biosciences. During the webinar, if you have any questions, feel free to type it in at the chat box on the top right-hand corner or in the Q&A tab on the bottom right-hand corner. We will have a live Q&A session at the end. I will now hand over the microphone to our marketing director, Dr. Jeff Xu.
Hey, good afternoon and good evening, everybody. Thanks for coming to the co-sponsored webinar by Precision Bioscience and ASEA Bioscience. This is Jeff. I'm the Director of Global Marketing from ASEA Bioscience. Let me give you a little brief introduction of ASEA Bioscience. We are a 15-year-old company based in San Diego. We manufacture instruments for measuring the cellular impedance and also another product line called Novosite as a desktop flow cytometer. We have more than 300 employees with more than 40 PhD and 29 distributors worldwide. Here is our development of the product line. First, come out with this, what we call the single plate system and the multiple plate system with a capacity from 196 well to 6 times 96 well. Later on, we introduced the DP system, dual purpose system, not only monitoring the impedance, but also the cell invasion and migration. In 2010, we introduced this high throughput model. You can use up to 4 times 384 well. Later on, we introduced the cardio system and the real-time cell analyzer cardio ECR system. Not only can monitor the contractility, but also can record the field potential for cardio safety purpose. Like I mentioned earlier, a little over 2 years ago, we launched this flow cytometer with 3 laser, 15 color, and 17 parameter. Okay. During the past 15 years, there are over 1,300 peer-reviewed publications citing this ASEA impedance-based real-time cell analyze technology. Our unique technology start with this E-plate. Let's have a close look at each well of the E-plate. Basically, there are gold-plated electrode at the bottom of each plate, covering almost 80% of that area. This figure actually explains how it works. Without a cell, the electronic current move very smoothly from the negative electrode to the positive. When you grow cell on top of the electrode, it actually resists and gives the impedance reading, which is converted to be the cell index reading. And when you have more cell on the electrode, or the cell grow bigger, or it has a morphology change, the impedance and the cell index change accordingly. So this is the basic theory how the technology works. Our system really provides a work-away operation. Imagine this: in a Friday afternoon, you have your regular, you have your cell from the regular flask. You scrape that cell and put on this E-plate. Then you can leave it in the incubator inside of the system, then go home. Came back on Monday, basically, you will have all the data, impedance data recorded, and it's a real-time time course. And at a certain time point, if you want to take that cell out and do some other experiment, it's okay, because this is really a label-free system. At the very right, that figure actually gives you an idea how your data will look like. When you Before the treatment, all the cells grow at the same rate, and when you give them the different concentration of the drug, the control still grow and reach plateau when it's 100% confluent with the cell, and with the increase of your new compound of the drug, the cell polymerization starts. That's why you monitoring, you can see the impedance, the cell index drop with a higher concentration of your chemicals. Here is the cartoon actually to let you know how our system can be used with immunotherapy. Basically, you can coat the tumor cell on top of the electrode and let it grow. It will, grow and reach plateau when it's 100% confluent. Meanwhile, the non-adherent effector cell will give very low background of this cell index reading. However, when you lay your effector cell over the target cell, the tumor cell, the cell, at a different concentration, low effector, different effector to target ratio, the killing starts. So that's why you can monitor the impedance change by looking at, by giving the different effector to target ratio. More recently, we actually not only using our system to monitor the tumor, solid tumor cell. We launched a couple kit. I think Dan will mention this during his talk, to study the liquid tumor by using different antibody to tether the B cell or lymphocytes to the plate, and then add the effector cell on top of that to monitor the killing. Our real-time cell analyzer can be used in multiple research areas, including drug discovery, cardio safety, cancer, immunology, wound healing, neurobiology, microbiology, virology, regenerative medicine. Regarding immunotherapy, there are lots of publications in different areas covering NK cell, T cell killing, bispecific T cell engager, antibody-dependent cell killing, and macrophage and complement mediated cytolysis. You can find all this publication from our website, the online publication library. Okay, finally, it's my great pleasure to introduce Dr. Dan McLeod. Dan got his PhD from UCSD and stayed a couple more years at this beautiful city for his postdoc training at Scripps Research Institute, then moved to North Carolina to join GSK. Now, Dan is in charge of the cell therapy research team at Precision Bioscience. My first meeting with Dan was at actually Washington, D.C. This past April during the ASEA-sponsored cancer symposium. Dan is a great speaker with lots of exciting data. Since then, Dan and I began talking about how could we have another talk to a bigger, broader audience instead of just existing accepting user. So today is a perfect opportunity for Dan to give a talk about how to optimize the design of CAR T cell. And thanks for Vivian and Dan. Let's give back to Dan now. Thank you.
Hi, Dan. I'm going to give you the screen now.
Okay, great.
Okay. All right. I can see your screen perfectly.
Okay, great. Thank you, Vivian and Jeff and everyone else at ASEA Biosciences for organizing this webinar and for inviting me to share some of our exciting data. And thank you, Jeff, especially for that nice introduction. As Jeff mentioned, today I'm going to talk about optimization of chimeric antigen receptor T-cell design guided by sensitive assessment of B-cell killing. And this really relates to our use of the ASEA Xcelligence instrument and some of the applications that Jeff described. excuse me, I'm losing my voice a little bit. I think scheduling a webinar was a good way to ensure that I would get a cold about 2 days ago. So I'm going to try and work my way through it, but I hope that everyone can hear me well. I'd like to start today by giving a quick introduction to precision biosciences and talk a little bit about our gene editing technology and our approach for generating allogeneic chimeric antigen receptor or CAR T cells. And then I'm going to give a couple of examples of so example one is using sensitive real-time B-cell killing assays using the ACA Excelligent system to really detect subtle differences in CAR T-cell function. And then I'll talk about the second example, which really built upon the first one, that we really started using the instrument in a different way. We realized the potential to use it as a screening tool, and it's really increased our ability to develop new CAR constructs and new products. So Precision Biosciences, as Jeff mentioned, we're located in Durham, North Carolina. We spun out of Duke University about 11 years ago. And we're a genome editing or gene editing company dedicated to improving life. We've been working on the technology for those 11 years and have developed a proprietary ARCUS next-gen editing technology. We feel it offers superior specificity and control over outcome and has a number of particular advantages. Conceptually, it's very similar for those of you familiar with gene editing, and I think everyone at this point has heard of CRISPR-Cas9, perhaps TALEN, zinc fingers, some of the other technologies. They all work in a similar way, and I'll talk about that in just a second. So we're using our technology in a number of different areas, and what I'm going to talk about today is immunotherapy, but we also have programs producing products for gene therapy, so using our technology in people and also in agriculture and food products. So very briefly, what is gene editing? Gene editing is simply the process of manipulating the function of a cell or organism by making a direct modification to the chromosomal DNA sequence. Gene editing really requires a precise endonuclease tool. Most people, again, are familiar with CRISPR-Cas9, and our tool and all the other ones work in a similar way, all the endonucleases. Essentially what they do is they're designed to bind to a very specific DNA sequence, your target sequence, and create a double-strand break in the DNA. That's all that the endonuclease does, regardless of which technology it is. The cell, when it has a double-strand break, that's a very bad event for the cell. It needs to repair that break before before any additional damage can occur, or potentially mutagenic events or serious mutagenic events. So it has a number of mechanisms for doing that. And which mechanism is used and other treatments that you perform to the cell determine what happens at that point. So after you've introduced an endonuclease and created this break, one repair mechanism listed on the left is a mutagenic repair mechanism called non-homologous end joining. Basically what that means is that on each side of the double-strand break, some of the nucleotides are chewed back to form a compatible end, and then those ends are stuck back together. What happens here is that the sequence is mutated, and most often if that's occurring in the coding sequence of a gene, you're going to disrupt that gene sequence and end up with a knockout. To generate either of the other incomes, either gene knock-in or gene repair, basically you have to trick the cell in a way and exploit a process for DNA double-strand break repair called homologous recombination. What you're doing in these scenarios is you're introducing a piece of DNA. In this case, it's shown as a plasmid DNA, but it could also be presented as a viral vector, as I'll talk about. And it carries those pieces of sequence that are shown here in green that are homologous, so they're identical to the sequences at the site of the double-strand break. And the cell would prefer to repair these breaks through homologous recombination with a sister chromatid. So if it's gone through S phase and started to replicate its DNA, it has a perfect copy that it could use to repair a double-strand break. That's the natural process. So what we're trying to do here is trick the cell into recognizing those green pieces of DNA that are annotated in green as the homologous piece of DNA to the site. So the cell thinks that it's repairing the break in a seamless fashion. And what it's actually doing is it repairs from each end and inserts the cargo at that site in the case of gene knock-in, or repairs a hereditary defect in the case of gene repair. So today I'm going to talk about gene knockout and knock-in on the left and in the middle. Quick background on our ARCUS gene editing technology. Again, it's very similar to what's listed in the middle box, CRISPR-Cas9, zinc finger talon in concept. It binds to DNA and creates a double-strand break. Where it differs is that this is actually a synthetic nuclease derived from a homing endonuclease, which is actually a natural gene editing system evolved for function in eukaryotes. It's very small. It's 930 base pairs. It's a single protein. It's unlike all the other ones listed here, which are multiple components. You can see from the size, it's about a fifth the size of Cas9, so it makes it easier to deliver. It also is very efficient at promoting homologous recombination. So this is very important for gene knock-in or gene repair. And this is because on the bottom left, you can see that there's a kind of a schematic of a 4 base pair 3' overhang. This is what's generated when our nuclease cuts a target sequence, and these overhangs are required for efficient homologous recombination. So we have an advantage there in terms of doing gene knock-in. The final thing I want to mention is that ARCUS is exclusively owned by Precision and is unencumbered by third-party IP, which is a very good place to be in the gene editing field. Briefly, chimeric antigen receptor, or CAR T cells, I think a lot of people in the immunotherapy space are familiar with them. It's a cancer treatment in which T cells are genetically modified to direct them to attack tumor cells. So the CARs themselves are synthetic receptors that combine an extracellular antigen recognition domain, most commonly an scFv, as it's listed in this picture, single chain variable fragment of an antibody, so you can recognize different antigens with that antigen recognition domain. In this example, CD19, which is expressed on B-cell derived leukemias and lymphomas. This antigen recognition domain is linked to co-stimulatory domain and CD3 zeta most typically, to enable proliferation, cytokine production, and cytotoxicity of T cells upon recognition of their target. And again, in this case, a CD19 positive tumor. So you're combining the antigen recognition capabilities of an antibody with the signaling capabilities and killing activity of a T cell to redirect the T cells. And there's a reason why we're working on CAR T and why it's becoming a much more well-known and well-established therapeutic approach. There have been some extremely successful early results in the clinic, in particular in leukemias and lymphomas. A number of examples, this one published in the New England Journal of Medicine, a lot of press coverage about really some patients that are at the end of their end stage. They really don't have a lot of other options, and really had no other choice but to try these experimental therapies, and really some dramatic results in terms of complete responses and remission of their disease. So I've done very well in the clinic. What most people, or most of the trials to date have done, is used an autologous approach. So the autologous approach is to extract T cells from a patient, engineer them, and then use those cells to treat that same patient. So a very personalized approach. The drawbacks, I think, are pretty obvious. It's expensive and difficult to scale. You have a delay due to manufacturing, and some patients actually do not survive long enough to receive their treatment. It's extremely variable from one patient to another, as you'd expect. It's a different person's cells each time. And not all patients are candidates due to the advanced state of their disease, so not everyone could be treated in this way. Our plan and our approach, and we're partnered with Shire for 6 undisclosed targets in the CAR T world, will develop allogeneic or donor-derived therapies. So we want to take cells from healthy donors, modify the cells, and then have a product that's off the shelf, readily available to deliver immediately to patients. So in addition to being immediate, it's cost-effective and scalable, consistent, and suitable for more people. In order to make a universal or allogeneic CAR T product, there's really two essential features, one additional one one in addition to just adding a CAR in order to make this a safe and efficacious product. And really to make it safe, we have to do this modification to knock out the T-cell receptor. So if we had cells from a unrelated donor and put them into the recipient that we were hoping to treat, and they had their T-cell receptor, they would recognize that recipient's tissues as foreign, and you would have graft versus host, which would not be a good outcome, obviously. So that's the first thing we need to do. Second, obviously, we need to knock in our chimeric antigen receptor to enable our T cells to recognize and kill the target cancer cells. And in our approach, I think what we're doing, which is really unique, is that we can do these two things at the same time. So we're exploiting this homology-directed repair approach to target the knock-in of the CAR transgene into the T-cell receptor alpha chain locus. So we're knocking in the CAR and knocking out the endogenous T-cell receptor at the same time. And this is a schematic of how we do that. So we use our nuclease to create a double-strand break in the TCR alpha gene. The nuclease is delivered to T cells by electroporation. We create that break. And what you can evaluate this by looking at expression of CD3, a component of the T-cell receptor complex. So a reduction or elimination of CD3 expression on the cell surface is indicative of knocking out the T-cell receptor. And you can see in this case, we had 55% of our cells were edited to remove the T-cell receptor. Taking that a step further, if we introduce our nuclease and also provide a repair template that has our CAR transgene and a promoter, and again, those regions of homology to the site in TCR alpha, this transgene and the homologous this transgene will become integrated at the site of the double-strand break as the cell repairs that break, and we end up with a defined integration of the CAR at that site. So we do knockout, knock-in at the same time, knocking out the T-cell receptor, knocking in the CAR, and you can see that the CAR is expressed, looking on the y-axis. And in our case, since any cells that still have their T-cell receptor could cause graft versus host, we magnetically deplete those cells to get a more uniform population. One thing I should mention, which is important for one of my examples, is that you see the CAR expression kind of appears as a shoulder and is not really a distinct population. And this is largely because the reagent that we're using to detect the CAR it's been well known to not be a very good reagent for detection. So it looks really more like a shoulder coming off the negative population. And that will lead nicely into my first example. So that's how we perform our knockout, knock-in approach. When we do this and evaluate the CAR T cells that are gene edited to remove the T-cell receptor, we see that they have potent cytotoxic activity in vitro against Raji and NALM6, which express CD19, but not against U937s, which do not. So they have specific activity. And this was using, importantly, an LDH release assay, so a non-radioactive CTL assay, very typical in the field. But it's usually a short-term assay, 4 hours or so, and you need high effector-to-target ratios to see a good amount of killing. That'll be important as I talk about our use of the ACEA Excelligence instrument and the real-time cell-killing assays. You can get to a much lower E/T ratio and look at longer periods of time. Additionally, they secrete cytokine when exposed to CD19-positive cells, so including interferon gamma, IL-2, and others, and not when exposed to CD19-negative cells. So they act as you would expect a CD19 CAR T cell to do. They respond to CD19-expressing cells, and they're also quite potent in vivo. So when we looked at a mouse model of using Raji cells that were, so a B-cell Burkitt's lymphoma cell line, expresses CD19, you inject them into NSG mice, allow them a few days to establish, and then inject CAR T cells. So we see in the PBS treated group, or the T cells that were only edited to remove the T-cell receptor, in those control groups, all of the mice are euthanized within about 17 to 19 days. And then we treated some groups of mice with 1 million, 2.5 million, or 5 million of our CAR T cells. And you can see on the right side, the survival figure, that all of the mice at the higher and medium dose groups survived throughout the course of this study, and even the mice in the 1 million cell, the low dose group, has significant survival advantage. And you can see this in the images, the bioluminescence imaging of tumor growth in the mice as well. And we were fortunate to publish these results in Molecular Therapy earlier this year, in case anyone would like to look at some of the figures I presented in more detail. So that's how we make our CAR T cells. And I want to go into the specific examples of how we're using the Xcelligent system, and how we're really excited about what it can do for us in terms of optimizing our CAR T design and moving towards the next generation of CAR T cells. So in the example one, what I'm going to cover is that we used sensitive real-time killing with the ACA Xcelligents to evaluate what were really subtle differences in CAR T cell function. And what we did in this example, so I mentioned the problems with the detection of the CAR using the standard off-the-shelf reagents. So what we did was we tried to incorporate an epitope tag into the CAR to potentially improve detection of our CAR expression and to allow enrichment of CAR-positive cells if we desired. And we evaluate killing of B cells by traditional methods and by using the ACA Xcelligent's real-time B cell killing assay. So again, I just want to mention that the commercially available reagent, a recombinant CD19 protein, doesn't do a great job of separating those cells from the CAR negative population. You can see that there are CAR positive cells, but they're not well separated. If you put in an epitope tag, and we put in a linear epitope from CD34 that's recognized by an antibody called QBEN10, if you take these same cells and then you stain using this antibody, you can see that actually you can now see pretty clearly separated CAR positive and CAR negative cells. If you look on the bottom right, clearly separated CAR positive cells when using this detection method. So we were pretty excited about that, and we could potentially use this to enrich those CAR positive cells. Miltenyi has a commercially available kit using this antibody to select CD34 positive cells. So it was exciting that we were able to do this. And one of the things we wanted to check when we made this modification was just to make sure that it wasn't affecting the function of our CAR T cells. So at first we were using this traditional LDH release CTL assay, which uses relatively high EDT ratios in a short period of time, so ranging from 40 T cells per target down to 1-to-1. What we saw when we looked at our original CAR that did not have this CD34 epitope tag, when we incubated with Raji or Dowdy cells, we did see killing, in particular at the high EDT ratios. And when we looked at the CAR that had this tag incorporated, it was a little bit of a mixed bag. It looked like the Raji cells, maybe there was a little bit less killing, but the Dowdy cells, maybe a little bit more killing at the higher EDTs, or at the low EDTs without the tag. So there really wasn't a discernible difference in the ability of our T cells to kill targets when the 34 tag was incorporated. And this was true at least by this traditional CTL that a lot of people in the field use. And about this time, we started doing a demo of the ACA Xcelligent system. And what we started using was this B cell killing assay that Jeff described, where essentially use an antibody, an anti-CD40 antibody. In this case, it binds to CD40 on the B cells. So you can tether your B cell targets, which are normally in suspension. You can tether them to the plate, and then if you add to CAR T cells, impedance is reduced as the CAR T cells are killing your target cells. So essentially, the cell index is used as a measure of proliferation and then killing once you've added your effector cells. And when we performed this assay with these two CAR constructs where we didn't really see a discernible difference in killing activity using the traditional assays, What we saw at a high ET ratio kind of reflected that. So if we did a 2-to-1 ratio, and this is just looking at cell index over time, and I should mention targets and effectors were added at the same time in this assay. I'll have some assays later where targets were added first and then allowed to bind and then adding effectors. But in this case, we saw that the Raji alone saw that cells grow until they reach confluence. Adding our TCR knockout control cells compete a little bit for resources, so the growth of the tumor cells is a little bit slower, but they also reach confluence at the later time points. And if you have a CAR with or without the epitope tag, at a 2-to-1 ratio, we saw essentially complete killing of the target cells. However, when we put the cells at a more stringent ratio, which you really can't do with those traditional assays because you don't see killing at those ratios over that time course, we actually were able to tease out differences that were not discernible at the higher E to T ratios. And this was very interesting for us. So now looking at the purple line, so at the 2-to-1 ratio, the CAR T cells with the 34 epitope tag incorporated perform just as well as without. But when you put them in a more stringent scenario where they need to proliferate more and perform better and actually are not able to overpower the tumor, they have to proliferate and perform serial killing, you see that there are differences and that the CAR with the 34 tag incorporated was not able to completely kill the tumor cells, and they're growing out at later time points. The other thing I should mention is we're using very low numbers of cells for this assay. So 10,000 target cells per well and 5,000 effectors. So just to summarize this first example, we modified our CAR construct to incorporate an epitope tag to potentially boost our ability to detect the CAR and then also potentially enrich for those CAR-expressing cells. We were pretty excited because we thought this would be a great approach. And killing of B cells by traditional methods didn't reveal any functional differences. However, once we put these same cells into a real-time cell killing assay using the Xcelligence instrument, it revealed that inclusion of the epitope tag actually did impair the function of the cells, and we're looking at other approaches now because of that. But we wouldn't have been able to detect that using traditional assays. So the sensitivity was really a key feature. Because we saw that it was such a sensitive tool, and because we saw that we really didn't need to use a lot of cells, we made the move to really start using this system not only to replace our traditional CTL assays, but also to take advantage of that sensitivity and use it as a screening tool for new CAR constructs. So in this example, I'm going to talk about is using real-time cell killing, using the leukemic cell killing assay, and how we use it to screen CAR T cells produced with a panel of different promoter variants to give a variety of CAR expression levels. So I discussed earlier when talking about the company and our approach, how we're doing this targeted insertion approach. So we're targeting our CAR transgene to a specific location. Again, this is very different from what most CAR T companies are doing. Most CAR T companies are using lenti or retroviral vectors. It's an efficient and proven method. But what it does is it delivers your CAR transgene, but it inserts it randomly in the genome. It could possibly be in several places. There are a number of issues with that. Number one is that random insertion means that depending on where it inserts, it may cause different levels of expression on the cell surface, so you end up with a very heterogeneous population. Our approach is much more consistent, and expression can be much more easily tuned, as I'll show in a minute. The other thing is that during development, if this is your approach, you need to produce a viral vector for each individual CAR construct being evaluated. And this really limits your throughput and what you can evaluate. We've actually developed a proprietary non-viral system. This system saves time, but it's not quite as efficient. So we only get about 5% CAR-positive cells compared with about 50% CAR-positive when we use our system with AAV or other viral vectors. So it's lower efficiency. And when your CAR-positive frequency is so low, it can be very difficult to evaluate the function of those cells. So you really need a a sensitive system, and that's where the ACA Accelogen system came in and where we saw that this sensitivity could be an advantage. So comparing our non-viral method and our method using AAV, the non-viral method on the top, so on the top left is just our TCR knockout control. So we have CD3 negative and CD3 positive indicating our knockout cells and cells that are unedited. Using our non-viral method, you can see that there's a low frequency of the CD3 negative cells express the CAR. It's again, we're usually in the 5 to 10% range. So it's a low frequency, but it's quick. We can screen a lot of constructs. The problem is, again, if only a small proportion of them are CAR positive, it's really hard to characterize those cells. In contrast, on the bottom, using our AAV method, you can see on the bottom right, it's a very high efficiency, almost half of our total cells, and more like 60 to 70% of the TCR knockout cells are expressing the CAR. So this is really useful for further characterization and for product development. But again, you need to make a viral vector every time you want to produce one of those. So with our non-viral method, we decided we wanted to test out how this would work and if we could take advantage of the sensitivity of the ACA Accelogens instrument to evaluate this in more of a screening method. So for this assay, we actually switched to using the leukemic cell killing kit. The reason for this is that, as Jeff mentioned, it tethers cells with an anti-CD29 antibody that works well for K562s. We had also generated K562 cells that express CD19, which we call K19s. So we have matched controls, a parental cell line and the same cell line that expresses the target that we're going after with our CAR T cells. So in this assay, it's very similar to the assay that I just described, but instead we're using K562s or K19s tethered to the plate and adding our CAR T cells and looking at the cell index as a measure of proliferation or killing. So going back to our targeted integration approach, one of the huge advantages that we have is that since we're targeting the integration to a single point, we're really able to produce a more homogeneous population of cells. The other thing that we're able to do, since we know that we're targeting it to one site, is we can actually change the promoter, and just by changing that, we can change the amount of CAR that's expressed on the cell surface. And this is something that you really can't do with a lenti or retrovirus other than by increasing or decreasing the amount, and then you still have very heterogeneous amounts of CAR because different cells will have different numbers of integrated copies. So we have the same number of integrated copies of the CAR and just dial up or dial down CAR expression. And so we actually engineered some proprietary synthetic promoters, and we can very specifically control the amount of CAR expressed on the surface by using these different constructs. And again, the low frequency of CAR-positive cells in this experiment is because this was done using our proprietary platform that does not use viral vectors but is lower efficiency. But very clearly, you can see that we have CAR T cells expressing relatively low, medium, and high amounts of CAR on the cell surface. And we wanted to put these CAR T cells into this assay to see if, even though they're low frequency, if we could detect differences in function between these promoter variants. So when we put these cells at a 1:5 effector or CAR T to target cell ratio in the ACA Accelogens instrument, what we did was we seeded the target cells first, allowed them to adhere for a few hours, and then added our effector cells. We see a little bit of a dip after adding the effector cells. I think that's just a little bit of disturbance of the tethered cells, and then they kind of recover and grow back over time. These are K562 controls, so they do not express CD19, and we don't see any significant decrease in the cell index, where cell index is really a measure of the survival and growth of the target cells, and the inverse will be the cytotoxic activity. However, when we looked at our K19 cells, the K562s that express CD19, not only did we see, were we able to detect killing with our constructs, we're able to detect very slight differences going from the low to medium to high expressing CAR constructs. And again, this is using only 2,000 effector CAR T cells per well. So our total cell numbers end up being higher because of the low frequency. So it's about 100,000 total cells. Only 2,000 of those are expressing the CAR, and we're still able to detect the activity of those cells. So it was quite impressive to see that even with a very low frequency of cells and a low cell number, we're able to detect really subtle differences between low, medium, and high expressing CAR T cells. Importantly, we want to verify these results seen in our screening platform, the non-viral method, with cells produced with our somewhat more involved and somewhat more difficult to produce AAV method. So I'm just comparing here on the top panel. You can see again these same cells, the TCR knockouts and the low, medium, and high expressors produced with the non-viral method. Again, we get a low frequency of CAR-positive cells, but it's quick. We can screen a lot of constructs. It's not feasible for really further characterization or use as a product. And this is in contrast to the bottom panels using, again, the low expression, medium expression, or high expression constructs that just have these different promoters. When we use the AAV method to target the integration, it gives us a much higher frequency of CAR-positive cells, and that can be used for further characterization and product development. But again, you don't want to do that for every single CAR construct you want to test if you can avoid it, just because making an AAV vector for each is expensive and quite time-consuming. So we wanted to compare the non-viral and the AAV method, or cells that were produced using these methods. And when we looked again in the ACA Acceligence instrument at these, either the TCR knockout controls or our low, medium, or high expressing CAR T cells, the trends were very similar. So there was a little bit, some cell differences in the cytolysis that we're seeing at a 1 to 5 versus 1 to 4 ratio. But overall, we can see the very similar trends, and we're able to detect the same differences using our more therapeutic platform, the AAV method, with our very quick non-viral method. So we're really excited to see that, because this indicates that we can test a lot of constructs without making viral vectors. And even if we're only generating a couple % of CAR-positive cells, we're able to screen the function of those cells by exploiting the sensitivity of the ACA Acceligence instrument and the real-time cell-killing assays. So in summary, our proprietary system for generating a low frequency of CAR T cells very quickly and without the need for a viral vector really requires a sensitive system for evaluating CAR T function at low E to Ts. And you could never evaluate the function of these cells at such a low frequency with traditional CTL assays. And results, importantly, from our initial screen using our higher throughput non-viral method correlate very well with the differences that we see when we translate that to our higher efficiency and more therapeutically relevant platform for generating CAR T cells. And the results from the study, so leveraging our platform for targeting the integration of the CAR at a defined site, we were able to generate synthetic promoter constructs and make CAR T cells that express the CAR at defined levels. And in this particular example, increasing the expression level of the CAR enhanced the killing of CD19 positive cells. This is important because we might be able to generate more potent cells in this way. But the other way is important too. So being able to tune down expression of the CAR is important for other antigens that may be expressed at higher levels on tumors, but also expressed on normal tissue and where on-target, off-tumor targeting is a concern. So their tuning CAR expression might be able to enable discrimination of tumor cells from healthy tissue. So it's really an important advance. And our overall summary was that our gene editing technology can be used to generate potent allogeneic chimeric antigen receptor T cells. And several modifications to CAR constructs are really potentially of interest for improving CAR function. I think there's just a ton of things that can be done in the CAR T field, and a lot of things that people are trying. So one example I gave was the addition of an epitope tag for detection and enrichment. There are ways to optimize that. The other example that I described was using different promoters to optimize the expression level for your particular application. Beyond that, using novel signaling or co-stimulatory intracellular domains. A lot of people have looked at using 41BB, CD28, but even moving beyond that there's a lot of room for optimization. And certainly new antigen recognition domains to recognize different targets. So everyone is looking at how to get beyond CD19 and hematological malignancies. And furthermore, really exciting, I think, is other modifications to CAR T cells. So additional knockouts or knock-ins to improve their function. And all of these potential alterations can change the activity of the CAR T cells. In our case, we really like the sensitivity of the Xcelligence instrument because it will enable us to rapidly screen new CAR constructs, and it's really become a critical tool in our CAR development pipeline. So it's something that we're going to be using quite a bit moving forward as we optimize some of these different elements and others of our CAR T cells. So I'd like to thank everyone at Precision Biosciences who helped with generating some of the data that I presented today, specifically Bruce, Mark, Clayton, Joanne, Audrey, and Wendy. Did a lot of the work that I described or were involved in that work. I'd like to thank people at ACEA, including Debbie and Fabio, for helping us get our instrument set up, and Yama and Biao and others for helping us optimize some of these assays. We had some collaborations with UNC to help us set up our AAV production and mouse system and the mouse model. And we had some early conversations with Nabil Ahmed and some people from his group at Baylor, and to discuss their use of the Xcelligence instrument and optimizing some of these assays. And I should mention again that we're partnered with Shire for 6 undisclosed allogeneic CAR T products. So thank you again, everyone, for joining the webinar and for your attention. And I'd be happy to take a couple of questions from the Q&A session.
All right. Hi, Dan. I have a couple of questions from the audience. The first question is, it has been well established in the CAR T field that in vitro cytotoxic activity does not predict in vivo activity. How do you think the results you are seeing in these assays translate to the ability of CAR T cells to expand, persist, and control tumor growth in vivo?
Yeah, I think that certainly the in vitro cytotoxicity in some of those standard CTL assays that people use, either chromium release or LDH, as I mentioned, I think it really is a poor measure of the CAR T cells and their ability and how they're going to act when you put them in vivo, either in mice or in people. And the reason for that is that in those assays, you're using high E/T ratios. The more differentiated effector cells are certainly better killers in that assay. And those cells don't perform that well in vivo. So they're good killers in those assays, but you put them into mice or you put them into people, and they just can't expand and perform as well. I think in general, the field is moving towards using those, using CTL assays where you can do a lower E/T, so lower effector to target ratio. And that in those cases, you're able to, you're putting the cells in a situation where they not only need to be able to kill target cells and just overpower the tumor, but they actually have to expand and exhibit some serial killing. So in those cases, you're measuring parameters. You're not just measuring cytotoxic activity, you're measuring those other parameters which are really important for in vivo activity. And we're certainly still evaluating these constructs, but largely we believe that the data generated in these types of assays at low E/T ratios correlates much better with the in vivo activity of the cells when you start putting them into these mouse studies and into people.
Okay, and then another question is, how has the ability to use the Xcelligence instrument as a screening tool changed your approach to CAR T development?
Yeah, it's really had a large impact on how we approach our development program. So as I mentioned, just being able to screen more constructs when you don't have the cost and time associated with producing viral vectors, even at a research scale, that having to produce a viral vector really limits the number of constructs we could test, and it takes several weeks. That's the other thing too. Once generating a plasmid or a piece of DNA, it takes not a lot of time at all. But generating a viral vector takes several weeks, costs a lot of money. So, I think in terms of CAR T development, some of it has involved rational design, but really to date, a lot of the parameters for designing new CARs that function well and for what makes a potent CAR T cell, they haven't really been that well optimized to date, and they're kind of hindered in the field by the throughput when you're having to generate a viral vector every time. So anything that increases the ability to screen constructs and use fewer cells and get information that turns out to be relevant when you actually are using a viral vector and generating more of a therapeutic level cell. I think that really helps with development and testing a larger number of constructs and a lot of new things. So it's really changed the way that we look at our development programs.
Okay, and I think we have time for one more question, but if anyone else has any other questions, please feel free to type them in the Q&A or the chat box, and we will email you those questions later. Okay, and the last question, let me see. Are you planning to develop CAR T cell for treatment of solid tumors? And what additional modifications do you think would be the most important to translate the success of anti-CD19 CAR T cells to different antigens?
Yeah, it's a difficult question. I think everyone in the field is looking to try and translate the success that's been seen with CAR T cells for hematological malignancies to other indications, including some different solid tumor targets. And I kind of touched on this at the end a little bit, but I think additional modifications will definitely be required in the solid tumor arena, because I think there's both some potential knock-ins, so potentially allowing your CAR T cells to express additional immune modulators to promote a more robust immune response, and also additional knockouts, for example. So several groups have suggested that additional modifications to the T cells, like for the example, some people have suggested knocking out PD-1 or combination with PD-1 or PD-L1 inhibitors might be helpful. So I think there are a lot of additional modifications that could improve efficacy. And we're certainly hoping that a lot of these things could potentially be modeled in a system such as the Excelligent system. So I think you could imagine using, for example, PD-L1 expressing target cells, and then measuring the ability of CAR T cells to kill those PD-L1 expressing target cells, either with a combination treatment with a PD-1 or PD-L1 inhibitor, or in T cells that have been further modified to remove PD-1 expression, as others have described. So I think there's a big opportunity to make some additional modifications and evaluate which ones will be the most effective for that particular indication.
Okay, great. Okay, I think that's all the time we have for today. Any unanswered questions will be answered via email. And thank you, everyone, for attending, and a special thank you to Dan for presenting. Have a great day.
Thank you.
Thank you, Dan.
Thanks.
I will be in touch with you soon. Yeah.
Great. Thanks.